So in today's experiment, we are going to perform the microscopy and transverse section of the cassia bark. Cassia bark, as you know, is a very well-known traditional spice which is used by the entire people of Indian subcontinent. Locally, it is known as dalchini. In today's experiment, we are going to learn how to take the transverse section of cassia bark. Here you can see this is the cassia bark which is having characteristic aroma. Let us see how we are going to take the bark. Cassia bark itself is very hard in nature. So as such, if we take the transverse section, it is not possible. That's why at least you should boil it for 1 hour before performing the experiment. So we have already boiled the cassia piece, and here you can find this is the boiled piece of cassia bark. Take the boiled piece of cassia bark, then you place it on a slab. In this fashion, as it is very tough and very easy to hold. So you just hold it like that. First thing what you need to do, you just level the surface. You can take the transverse section in this way or this way also. Look it carefully how I am going to take the transverse section. Here you just remove the upper, just to level the surface, cut the little quantity of the topmost part. Then the surface has been leveled. Now with the edge of a sharp blade, always use new blade for taking the transverse section. With the help of the edge of a new blade, you cut very thin section of cassia bark. Here I am taking very thin section of this bark. Few of the sections already I have taken. Now little more I am going to take. I have taken few more. Now transfer into the water. Now from this you find which one is the thinnest section. Always remember, lighter the color, thinner the section, darker the color, thicker the section. So from that you have to take the thinnest section. So I have chosen the section and have placed it over a slide. Now what you need to do, first you add 1 drop of chloral hydrate, 1 or 2 drops of chloral hydrate. Chloral hydrate is used as cleansing agent. What it cleans? It cleans whatever the pigment, dirt, adhering substance, earthy substance, whatever is present, it helps to remove that. So just you add the chloral hydrate and wait for some time so that the coloring pigments goes away. After that, to the sample, you add 1 drop of fluoroglucinol solution and 1 drop of concentrated hydrochloric acid. So mix the mixture properly. Fluoroglucinol and concentrated HCl 1:1 ratio is used as staining agent. What it stains? It stains the lignin containing cellular characters to pink color. Now your slide is almost prepared. The reagents we have added. Now place a cover slip over it. Take the cover slip and place it. So take care so that air bubble should not be there. If air bubble is there, then remove the cover slip and place it once again. With the help of a filter paper, clean the base, wipe out the excess of liquid, and before that I have forgotten, one thing, just I am taking out the slide once again. Add 1 or 2 drops of glycerin in order to prevent the drying. Your slide is ready now. Now you place it under the microscope. Let us see under the microscope what we are going to get. Always focus on the 10x magnification power. So I have focused already. So here what you are going to observe, first thing, the topmost part is known as cork. You will find dark brown color cork. Below to that you will find the oil glands, which contain volatile oil, due to which Cassia bark possesses characteristic aroma. Then the cortex region. Cortex region consists of the medullary rays mainly, and several phloem fibers. Then in the middle part you will find a very characteristic pink color characteristic band. This band is known as sclerenchymus band. This sclerenchymus band made up of stone cell. What is stone cell? Stone cell is a rectangular shape of parenchyma cell, which the cell wall of this cell is made up of lignin. So like the brick, one after another, while making the wall, we are attaching the bricks one after another. Exactly in the same fashion, here also you will find. So many stone cells are attached here, and that's why, due to the presence of lignin, the entire band will appear as pink. So these are the certain important characteristic cellular feature of Cassia bark. So from the topmost, I repeat once again, you will find cork, then you will find sclerenchymatous band. Sorry, not sclerenchymatous band. It is called stone cell layer. Stone cell layer. After that, you will find oil glands. In the cortex section, you will find medullary rays. So that's all from the transverse section of Cassia bark.